So this talk is more about an overview of a program that we have just started. It's about a year old, and this is about how we can use computing for curing cancer. Of course, that's a big effort, but it's not something that we are taking it lightly. And this is a program that's a joint program between the Department of Energy and the National Cancer Institute, which is trying to build a program towards adapting high-performance computing solutions for the US with respect to what we can do for cancer. Right? This is a team that is fairly large across multiple different labs. There are 6 national labs that are involved, DOE national labs, which include Argonne, Frederick, Lawrence Livermore, Los Alamos, the National Cancer Institute itself, and Oak Ridge National Lab. If you have any interest in talking and knowing more about the programs, I would refer you to one of my colleagues here, Jim Glossley, who's right here in the front. And any tough questions are for him, the easy questions are for me. Okay? So I've highlighted his name too. All right. So why did we get into cancer research in the first place? So the aspect is that the DOE is something that is very much responsible for the National Strategic Computing Initiative. And what do we mean by that is that it is for transitioning HPC research into things that can be deployed for real-world purposes. And the National Cancer Institute is really interested in finding out advanced solutions for cancer. And you might have heard of the Moonshot Project and things like that. There is a precision medicine initiative and so on. And all of these things are focused on creating what we call as a cancer knowledge network that will eventually generate all of the future knowledge that we need in terms of scientific discovery and guide treatment. So this is really about pushing the frontiers of how we can develop novel solutions for solving cancer-related issues using computing as a platform, right? And there are 3 pilots that have been identified, and the pilots actually come from 3 different scales of problems that we look at. So the first pilot is what we call as the RAS signaling pilot. This is trying to understand the basic biology side of the story, which is to say, how does RAS, which is one of the key signaling molecules in cancer, involved in trying to start signaling processes that are related to cancer. So the idea is that RAS as a protein is something that people have studied for over 30 years. There are no targets for this particular protein. That means that it is almost undruggable. And so we want to tackle the challenge of understanding what does RAS do in the membrane. And this is combined with this aspect of saying, okay, we are going to try and run molecular simulations at scale. This is something that the DOE labs do very well. And we can actually scale these simulation softwares on our existing platforms. But the question is, the amount of knowledge that you're going to gain by just running the simulations is only minimal. So we want to drive that through machine learning and trying to understand what exactly does the RAS do in terms of its membrane function. Now we are going to elevate this problem from the molecular scale onto something where we can look at cellular signaling. So we want to understand more about the aspects of how cellular signaling happens. And for this particular reason, we are studying what are called as patient-derived xenografts. The idea behind that is you take a tumor that is taken from a patient, you directly inject that into a mouse, allow the tumor to grow, and then study the effects of drugs on these tumors and try to understand how we can do these experiments automatically. In the sense that the prediction part is about designing novel experiments that will help us probe better in terms of what experiments need to be done. The final pilot is the one that we lead at Oak Ridge National Lab, which is something that we have been doing for a while now. It is about the integrative modeling and analytics for what we call as surveillance of cancer patients. And here the problem is at the population scale. And the idea behind that is now you have electronic medical records for all these patients, and we would like to do automated natural language processing so that we can understand the text, extract context, and understand what sort of treatments are being done for these patients and how can we track them over time. And at the bottom of all these things, as you have seen, there are machine learning challenges that are ingrained into it. One of them happens to use unsupervised machine learning, which is for the molecular scale. We use supervised machine learning for the cellular scale of the projects. And for the third one, which is the population scale, we are using semi-supervised models because these are better suited for text-like modeling. And in each case, we also have an underlying modeling and simulation challenge that's being tackled at different scales. For one, I was trained in molecular dynamics simulation, so I'm going to explain a lot more about Pilot 2 today and the challenges that we face. But I'll also talk a little bit about the data challenges that we face across all these 3 different pilots and give you an overview of some of the treatments, some of the strategies that we have been building to tackle the data challenges, right? So underlying all of these, this is a pilot that's gone all the way to the DOE headquarters, which means that Rick Perry, who is currently the Energy Secretary, knows about these projects, and he's quite supportive of all the things that we are doing. And from the other side, we have Doug Lowy, who is acting director of NCI, who is pretty much involved in all these 3 pilots. You have seen the different 3 pilot projects. But at the bottom of it, because we are handling challenges, data challenges at scale, we are looking at really building machine learning tools that will scale to the next generation of supercomputers that we are building. So if you know, Titan is the current supercomputer at our lab. We are going to replace it with a new computer, which is called Summit. A similar change is going on with Livermore, where they're going to replace their current machine with what is called Sierra. And these computers have computing power that is unprecedented. So we really want to take and make these deep learning applications available on these systems, and therefore we are actually invested heavily in terms of building the next generation of tools. So let me talk a little bit about each of these pilots. As I go along the pilots, I'll try and explain some of the open questions and challenges that are out there, and then we can discuss a little bit about what we are facing. So okay, I'm not going to spend a whole lot of time on Pilot 1. And Pilot 1 is essentially about building predictive models for preclinical screening. And what do we mean by that? Is that we have all these cell and compound-based screens that are available. So you have compound data sets. That means these are drug-like molecules for which there are very critical characters that have been listed for, which means that each compound is described by, let's say, a 6,000 or 7,000 feature vector. And the question from that is saying, using those 7,000 feature vectors, and using a tumor model that we have been building it on a mice, can we actually understand whether that drug has an effect on the tumor or not? It's a very simple classification problem in some sense, but the problem is that you're dealing with millions and billions of drugs. So if you take the small molecule library, there are more than at least 4 million data points available, and assuming that you have 4 million by 6,000 or 7,000 descriptors available for them, now you can see the problem, which is very compounded. And a similar way, if you look at the patient-derived xenografts that are coming from mice, you have the same level of heterogeneity because now the mice are being implanted with the tumors. There is tumor heterogeneity, which is intrinsic. So you have different cell lines. You have all these combinations that are happening. So therefore, this problem is really not that easy to tackle on just your laptop or my laptop or anybody's computer. So the idea behind that is we want to take all these cell lines as well as the compound databases that are available, and you can see in each case we have marked up how much data sets or how many terabytes of data is already available, and in some cases these data sets are even more than several petabytes. So when you have such a situation, you have really a complex amount of information available to us, but we really don't know what sort of features are important, whether we can use these features for doing analysis, whether we can do them for optimal experimental design. So there are a few open questions here, which is how do you represent drugs, which is something that is of potential interest to me, and how many representations are necessary and sufficient to capture the sort of information that we want? And can we predict all the qualities that we are interested from a given drug just using its structure? For example, if you have a SMILES descriptor for it, can we predict all of the properties that are needed for it? The second aspect of it is concerning how do we design experiments using these types of information. So there are active learning techniques that are available, or reinforcement learning, where the ultimate goal is to say that you want to find the drug that has the maximum efficacy, but also worry about safety and things like that, which are of deep concern. The second pilot, as I have told you previously, this is about the RAS signaling on the membrane, and this actually concerns the first level of complexity that we are building into the modeling and simulation guided whole. So the experiments are coming from the NCI itself, which is Frederick National Lab, which is doing a lot of the experiments. That includes cryo-EM imaging. ORNL is doing a lot of the X-ray and neutron scattering. And we have multimodal experimental data that is actually coming through to us at a very large scale. Now the problem is, if you want to try and understand what's going on in a cell membrane, especially with this RAS bound to the cell membrane, there's even the simple aspect of understanding how RAS interacts with the membrane, which itself is not known. So one thing that we can do is go back and say, okay, let's simulate this using molecular dynamics simulation. The great thing about molecular dynamics is that you get all these three-dimensional coordinates, you can track these particles, you can understand what's going on, but your experimental observations that you have are extremely sparse. So you need to build new types of machine learning tools that will allow us to kind of weigh some of these things automatically so that we can say, okay, this might be a possible hypothesis for the observation that we are seeing in an experiment. So that is one angle. The second angle is that some of the events that we are observing at scale for these types of experiments, they occur at seconds, milliseconds, and timescales like that. So now your feasible MD code might not really run at those timescales, and so we want to develop new adaptive sampling techniques that allow us to scale to these molecular events. So if you really look at the problem of molecular dynamics, it's not about the ability to generate the data. Right? People have been very successful in terms of generating the data. You might have heard of Anton and other machines that are doing this at a very regular scale, where you're getting up to millisecond timescales of simulation. That's not the problem. But the real problem starts with the amount of data that's being generated. So let's say I have a millisecond timescale simulation of even a very small protein. This can exceed several terabytes even on a simple system. And therefore, given that data generation is not a problem, the analysis becomes the key bottleneck in terms of saying what is going on. So we think of these events in 4 types of ways. So the machine learning has to really focus on understanding these multi-scale simulations, where you have to understand events that happen at nanoscale, and there is time scales associated with each of these events. For example, when the RAS molecule is bound to GDP, it's in an inactive state, then it binds to GTP, it becomes an inactive state, but these events happen at microsecond timescales. And then in order to look at what happens when 2 or more molecules interact with each other, now you're bringing these aspects of saying, okay, I really cannot simulate at all atom scale all these different large scale systems. So you need to move from all atom to coarse grained models at some point. And that doesn't happen automatically. You need some sort of guides to do that. And then you can take it further down into some models which are called as continuum model. I'm not going to describe these in details, but you're really capturing these multi-scale behaviors at every time step. And therefore, you need sort of, to transition between the all-atom and coarse-grained models, you need tools that allow you to do event detection. That is, you need to find time points where there is deviation from normal behavior. The second aspect is you also want to say whether have I seen something novel about these states. So a protein undergoes many of these state transformations. We would like to understand what's going on with respect to these states, and therefore there are ways to do that in an unsupervised way, which is to cluster and visualize the data. So the clustering and visualization must respect the basic biophysics of what you're trying to understand, and that is something that we are definitely building. The third aspect is that once you see the data, you also want to build it in certain terms of feature extraction, which means that it follows the intrinsic statistical details of the data so that you can do this in an automated fashion. And the last thing is, because you have sparse experimental observables, you need to merge the two somehow. You have these high dimensional data sets that are coming from MD simulations, and you need to merge that with experimental data that is potentially really sparse. So you need some sort of Bayesian inference frameworks that allow you to interact with these sparse experimental models so that you can integrate the information across multiple scales and start saying something meaningful about these systems. And of course, there are deep learning equivalents for all these 4 that we have to build. I won't talk about that today in that much detail. So let's take an example of an event detection algorithm. So the motivation behind this is, let's say this is a protein structure. I have all the atoms displayed like that. And if you treat the entire protein as a movie that's evolving over time, you're tracking these Cartesian coordinates that are evolving over time until time t. The questions that we are interested in saying is, are there regions that are moving together? Right? I might have some regions like this that are highlighted in red that are moving together. I would like to find them because they represent where the binding might happen. Or there are also time points where these events happen. So at T0, you can see the gray line which is shifted, and then at T100, it goes all the way down, and then at T6000, it comes back to some sort of an intermediate state. So you have these temporal dependencies also that you want to capture. So a lot of work that has been done so far has been using what are called as bag of conformers approaches, which means that you use PCA, you eliminate the second order correlations, and you try to say something about what is going on in the system by measuring the covariance between the different particles in your system. But the problem with PCA is that it doesn't really describe the spatial and temporal correlations simultaneously. You just have access to the correlations that are there with respect to the particles, and it's a very rough way to capture what happens in a system. So in order to do this, you need to find patterns with multi-way data, and the way you represent that is to use something called as tensors. And this is a conceptual representation of this. You can think of the data as collection of matrices, and over a time scale, you essentially build a multi-scale representation which captures n cross t, where t is the time scale where you're interested in particularly tracking this. The tensors are essentially n-dimensional matrices. They are not the stress and tensors that you observe in physics, but they are equivalent representations of the data. And then without getting into the details, we can actually build an anomaly detection algorithm based on this, which is based on minimizing the Frobenius norm between past and current observations. And once you do that, you can use this as a measure to actually identify where time points are changing their spatiotemporal behaviors. So this is an error of reconstruction plot that's been spotted over 180 time windows, and the corresponding same metric that you use for measuring whether your MD simulations are stable or not, which is called as root mean square deviation, is also shown here. But I want to identify 2 points that are shown in red dots there. And what you observe from there is that the structural difference that you observe between 2 points, one is shown in green, the other one is shown in gray, is that the structural difference is not that much. If you really look at the RMSD value, it's actually quite low. But if you observe the orientation of where the substrate is bound, it's actually changed a lot. And those sort of changes that you find in these systems is actually very important, because without that, you cannot say something about whether this is an important event or not. And so a lot of the work that has to go into doing these sorts of things has to deal with building event detection tools that can do this as the simulations are running. There's also this aspect that if you look at the data from biomolecular simulations in general, you observe that these data sets are heavy-tailed. So heavy-tailed distributions are very common across different applications. You see them in videos and images. You also see them in biomolecular simulations. In this case, what we are tracking with respect to a particular atom, I'm standing at an atom and saying, how much do I fluctuate with respect to the mean position? And if you plot a distribution of that, you essentially see that they have these long-tailed distributions. So a long-tailed distribution obviously means that you cannot use techniques such as PCA. So this is an example where PCA doesn't hold very well. Let's say I'm tracking 2 particles, glutamine 2 and leucine 14. These are the 2 particles. I'm just plotting the positional deviations with respect to each other. You can fit that with an ellipse. That's what a PCA does. The 2 arrows show where the principal directions are. But let's say I take these 2 residues, for example, glutamate 31 and alanine 45. And now you start to see the problem, which is that there are points in the gray which are actually from the data, which is not noise, which is something that tells you that there are intrinsic non-orthogonal orientations in the data which you must capture in order to do this sort of analysis. So we built some of the first algorithms to do this using higher order statistics, what we call as quasi-anharmonic analysis, where instead of minimizing the second order correlations like PCA, we instead minimize the 4th order correlations, which is using kurtosis. So kurtosis is a measure of the 4th order statistic in the system, and you can actually do this. And what it allows us to do is, this is an example again in lower dimensions, and the black arrows are representations of PCA, whereas the red arrows are from QAA, quasi-anharmonic analysis. And what you essentially see is that the red arrows are able to capture these intrinsic non-orthogonal directions. So why is this important? You go back and look at the system, and you can actually capture the presence of high and low energy states. Okay? I'm not going to go into the details of where we went with this, but this has been applied on other systems like intrinsically resolved proteins, where we can actually put it together with experimental data, and we can also look at how we can solve these issues with respect to molecular dynamic simulations and integrate them with Bayesian-like techniques. So you simulate, you cluster the data somehow, and then you build it back in terms of comparing it with experiments. I'm not going into the details again, but we end up showing that we actually get a good description of kinetic aspects of the landscape. So we can actually say what states are moving, how big are these states, how much time it takes before it jumps between these states. And ultimately, we can go back and reconstruct the experimental data also, where we can say it fits well with a neutron scattering observation. So this is a measure of saying how good can we be in terms of building the same representation. The last aspect is to show that we can do this using deep learning also. Again, we are doing this in an unsupervised fashion. So we identify these conformational states. If you see on the left-hand side, there are these plots where we show the presence of multiple states. And in this case, what we did was we took the folding trajectory of a protein, which is called FS peptide, and this has been converted into raw pixel coordinates, which basically tells you contact matrices, which tells whether two atoms are nearby or not. And as we do that along time, we can actually spread out the data and use deep learning to actually build a low-level representation that tells us something very useful about the evolution of the system over time, right? And there are some open challenges and questions here in the sense that, okay, now you're building these tools for doing it on smaller scale systems, but how about doing it for these large scale systems that I talked about, RAS and other things? So in this case, we have to build a workflow that is capable of doing these analysis as the simulation progresses. And there is also the need to do data analytic frameworks without need to modify the underlying simulation code. If you were to modify the underlying simulation code, you run into these aspects of bottlenecks that you have to handle in a different way. So there is a need to build in situ analytics for feature extraction and evaluation. And the simulation scaling has to be essentially done independently so that we don't interfere with that project, that part. And the propagation that we are talking about in lower dimensions should also be done in such a way that we can incorporate them continuously in terms of what a good model is, what is a good fit observed thus far, and so on and so forth. So there are some interesting challenges that come out of that problem. The last part, which is pilot 3, is something that Oak Ridge leads. And in this case, we are very much interested in taking these data sets that are coming from the NCI SEER data. I'll talk a little bit about that. And how do we process free text to extract information regarding biomarkers and things like that? And how do we put together patient trajectories in a very specific manner that addresses some of the questions that we have in terms of basic cancer research? So in this case, the cancer pathology report processing pipeline is very complex. So there's a patient that goes to a pathologist, and the pathologist gives the diagnosis of analyzing a tissue specimen. So they take a biopsy, they do the analysis, then they write a report, what is usually referred to as a surgical pathology report. And the surgical pathology report is read by a certified tumor registrar, and the tumor registrar reviews this whole data with respect to the patient's medical record as well as the path report and puts them together in what are called as cancer registries, which are basically large-scale databases consisting of information. And each cancer registry has its own way of recording information. Some of them store the images from the patient's pipeline, some of them store only the text data, some of them have different ways to represent, some of them even store tables. So given this sort of information, we need to take that and integrate it into structured data like an electronic medical records and show this sort of information, which is that this patient comes from Kentucky, this is the ID, this is the record number, this is what the tumor is, this is what the site is, and so on and so forth. So this has to eventually go into the SEER database. Currently, less than 30% of this data actually is annotated and available. But they have plenty of reports, which means that they have more than 7 million reports that are currently available for just one particular year for analysis. And there is no way to actually understand this. So the need is that we need to abstract structured data from free text pathology reports, but manual abstraction is extremely time consuming and not scalable. And therefore the goal is to develop a scalable framework that allows us to do this automatically. The way we think about this is to represent how a person like me or you reads a document and tries to understand the document. So we do what are called as word level embedding, which is to capture importance of words. So you read a word, you put towards attention, which basically tells you how important that particular word is. This is based on deep learning. So if you're interested, you can talk to me later about what the model does. And once we do that, we filter them based on their importance. And then we actually build them with respect to sentences. Now we actually can automatically capture which sentences are important. You do the same thing again. And now this is a hierarchy of documents that basically allows us to say, with respect to a particular class of documents, how do I capture all of the important words and so on. Without going into details, actually what we call as a hierarchical attention network is the one that performs best even compared to convolution neural networks and so on. And this is something that we have been working for a while now, where we can show that the specificity of these documents are equally higher, so which means that these documents do very well in terms of understanding the documents. And in terms of CNN and Han, we can actually identify very quickly what are the most relevant words for a particular machine learning task, and therefore we can identify which sentences are important, which words are important, and that allows us to basically say, just focus on these particular sets of lines and we should be able to extract the information. So what we are trying to do with Pilot 3 is now take these information sets and integrate them with other types of modalities, including imaging data. That's ongoing work with University of Pittsburgh, where we are taking these algorithms for imaging and putting them together with pathology reports in order to say where the tumors are located and what sort of things we are seeing. There's also the radiological side of image processing, where we do it for breast cancer and types of things. This is an ongoing collaboration again with Oak Ridge. And there is also what we are developing as deep text comprehension tools for Candle project, and integrating them with a lot of these environmental, climate, and syndromic surveillance data, including ICD-9 codes and information that comes from IMS data sets and so on. And we are pushing that together in order to do what we call as precision phenotyping, which means that how do we understand a person's phenotype given with respect to all of these complex data sets that are present with respect to the data. So the last part is about cancer deep learning, which is the Candle environment that we are building. And here these are tools about algorithms that we are developing. So if you take a simple convolutional neural net like AlexNet, which has approximately 12 million parameters, even small networks require a large representation of parameters, which doesn't make much sense in terms of how do we understand how algorithms work for deep learning. On the other hand, if you just look at how the convolution operation works, and the idea behind that is convolutions allow you to capture the intermediate neighborhood information very quickly. And if you look at the last parts where you see a lot of color patches, they are actually meaningless features. They don't capture anything useful with respect to the convolutions. All you care about are the lines and edges that you really care about. And therefore, there's a way to actually reduce the number of parameters automatically. And the way we think about that is to implement these filters or tensor-based operations as low-rank tensor factors. And the idea behind that is very simple from linear algebra, where you have a d cross 1, which is the matrix that you apply. But instead of doing that, we end up doing d cross 1 and a 1 cross d cross 1 matrix multiplication, which ends up giving you the same type of result. But instead of having 12 million parameters, we can actually reduce it to less than 10,600, which means that you have an order of magnitude reduction in terms of the parameter space. And why is this important? If you think of how these large-scale machines are being built up, there are these different layers where the deep learning networks play a role. You have the underlying CUDNN libraries, which are fantastic because they allow you to implement some of these kernels that are important for function. But the problem is the kernels themselves have limitations in terms of how they can scale and so on. That is something based on our experience. And they are not very well suited for exploiting the memory level interactions and things that we have to do in order to scale them. And elevating that further, there are tensor operations or multilinear algebra libraries that are not even being built right now, so we are actually trying to do that. And elevating that further, we have the hyperparameter sweep engine and things like that that we have to build in order to build our tool sets. And based on our preliminary information and what we are trying to do, is actually a result from a low rank representation of our convolutional network, where we can show that we can reach the same amount of performance thresholds like traditional convolutional nets, but with a smaller number of parameters that we need to do. At the same time, we don't need to scale or we don't need to subsample the data. So if you're doing convolutional nets these days, you end up patching the data, which means that you take a big image and end up patching it with 256 by 256 samples and so on. In this case, we don't have to do that. We can directly feed images that are the size of 2000 by 5000 pixels. And this is particularly useful for scientific data sets where things are much harder because just the data sets are themselves so complex and they also have long range interactions. So in terms of challenges and open questions in this area, there are some necessary primitive kernels that have not been built for scaling across multiple nodes, what we call as distributed kernels. And that is something that we are very much interested in building and trying to test them. In order to enable development of new classes of deep learning algorithms, as you all know, deep learning algorithms really do not have a way to incorporate first order logic or logic relations, and therefore that is something that we are definitely interested in. We are also interested in making adversarial modeling that becomes more robust. It pays attention to more of the features of the data rather than being very blind to how it is being done. And also building the math foundations for deep learning, which includes tensor algebra libraries, the kind of uncertainty quantification that needs to go on top of these techniques and so on. And finally, what we are doing with our industry partners is also hardware acceleration. So these primitives need to be built in such a way that they can scale on multiple systems and so on. So with that, I would like to thank you all for the opportunity to speak here. If you have questions and comments, just email me. I unfortunately cannot stay for all the 3 days. I have a review meeting that I have to attend tomorrow, but I really look forward to talking to you via email, or you can talk and harass Jim Glossley. He's here for the next 2 days. And if you have any interest in pursuing internships at the lab, just email me. And there is also a data science PhD program that we have recently started. And if you're interested in pursuing graduate studies, please do apply for it in the future. Thank you very much.